Let's now talk about gene regulation or the regulation of gene expression. And one important thing that we're going to talk about here is the Lac Operon, right? So to get started, because we're talking about regulation of gene expression, we need to understand what it is. So the regulation of gene expression. So from the word, what do you think this is talking about? One thing, it is regulation. Regulation is basically controlling something. And here we are talking about a controlling of gene expression. So, meaning that for us to be able to understand the regulation of gene expression, we need to understand what gene expression itself is. Okay. So, what is gene expression? Gene expression is a process by which information, which is found in a gene, is going to lead to the production of proteins. So basically, that is a simple definition. Again, what is gene expression? Like you have something, but you want it to come out. That is a gene expression. So gene expression is basically the process, okay, the process by which information encoded in or the information that is found in a gene leads to the formation of proteins. So information in a gene leads to production of proteins. And by this time we know how we do produce proteins. The first step in the production of proteins, first we are having replication, right? I'm also writing the same way that I'm saying. So production of proteins. So what I'm saying is that in the process of producing proteins, we have got phases. The first phase is we are having DNA being converted into another DNA molecule. And then the DNA molecule that we have can be converted into RNA, and then that RNA is going to be converted into proteins. So since this is regulation of gene expression, it means that before we can produce our proteins that we want to produce at the end of it, we can control this step, right? So we can control this at any stage. We can either control it at this stage. What stage is this? This is replication. Or we can control the formation of this gene at this stage. What is that stage when you convert DNA to RNA? That is transcription. You can as well control the formation of proteins at this point, RNA to proteins. And what's that? That is a translation. So basically, this is what is being involved when you are talking about gene expression. This is very important. Okay. So, what are the things involved in gene expression? Regulation, replication, transcription, and translation. Again, when you look at the regulation of gene expression, the control of the genes that you are making, there are some things that you want to do. So, we've talked about gene expression itself. But remember, this is regulation. So, what do you think regulation of gene expression is going to be? So regulation of gene expression is going to include a lot of different mechanisms that are going to be used by the cell to increase or to decrease the production of a specific protein, right? So that is basically the gene expression. So let's get to write that. So we are saying that the regulation of gene or gene regulation. These are steps involved in controlling. Now, what I want you to understand here is when you are controlling the genes you are producing, the control can either be increasing the genes or reducing, right? Like controlling, let's say for example, you want to form hair, isn't it? Hair is protein. So you want to either increase or reduce the production of a specific gene. That is the regulation of gene expression. I hope that is clear enough. So I needed to stress a bit on that one so that we just get to understand. So regulation of gene expression is increase, increasing or decreasing production of a gene or production of a specific protein. Okay, so basically that. So what is the importance of regulation of gene expression? Why should we regulate gene expression? That is the most important question. Well, one thing that we need to know is regulation is significant because when you are regulating gene expression, you are actually preserving energy. You know, every time you are forming a gene, you're actually making energy. Sorry, you're actually using energy. So if you don't, if let's say for example, every time you are just converting your DNA to RNA and then to protein, RNA, protein again and again, you are consuming energy. So the importance of the regulation or the control, decreasing the amount of DNA, decreasing the amount of DNA that you are going to end up converting into protein is one thing, is that is what saves the energy, because in each step of gene expression you are using energy. Okay, another important thing is that you want to ensure that correct proteins are made when and where they are needed. That's very important. You don't want to be forming proteins anyhow. You want to form proteins only when they are needed, and not only when they are needed but where they are needed. Ensures that proteins are made when and where needed. Okay, so basically these two functions are very important. This is why we regulate gene expression. Okay, this is why we are controlling. It's not every time that we are forming our genes, so we are regulating them. So because of that reason, now we need to understand something that is known as the Lac Operon when it comes to the regulation of gene expression. The Lac Operon is very, very important in regulating gene expression. So to understand the Lac Operon, the first thing is we need to know what is the Lac Operon and what are the components of the Lac Operon. So that is where we are starting. Let's write it again here. Lac Operon. The lac here actually represents lactose. So it says lactose operon. So what is the lactose operon or the lac operon? Genes in our body, we can actually get to group them or classify them. Okay? So we can classify genes into two main groups. So genes are classified into two main groups. And what are the two classifications of genes? We have got the regulatory gene, that is one, and also we have got the structural genes. Very easy, but we really need to understand this. So regulatory gene and structural gene. Okay. So what is the difference between these two types of genes? Regulatory gene is going to actually make up the majority of the genes that we have, like 98% of the genes are going to be regulatory and only 2% are going to be structural genes. So regulatory genes, even from the word regulate, these are genes that control protein synthesis. Okay? Control protein synthesis. And the structural genes are the genes that are going to be coding for proteins. Okay? So that is basically the difference. Regulatory genes are controlling protein synthesis, while structural genes are not coding for any regulatory thing. Or the structural genes are the actual genes that are forming proteins. So this is going to be an easy one. That's an easy differentiation that we can give. Now, if we have got a structural gene, let's say for example we get a strand of DNA. This strand of DNA is going to have a portion that is a structural gene and one portion that is a regulatory gene. Okay. Now a gene is basically a portion of the DNA that is going to control characteristics. For example, if I want to form proteins, for me to form those proteins, information has to come from a certain segment of DNA, and that's what we are calling as a gene. We are going to be doing genetics just after this topic, so you understand everything about genes, chromatid, chromosome, and everything else. So let's say this portion of this DNA is telling us we need to make proteins. So this portion we have got the structural gene. So this gene is telling us what? It's telling us, okay, bro, you have cut your hair. You need to make new ones. And so you are going to utilize that. But then before this structure gene can start to form that those proteins we're talking about, you have to control them. You have to tell them, structure gene, can you please make some uh, proteins? Can you give us some information to make some proteins? So you have to tell it. But who tells it what to do? It is a regulatory gene. Okay, a regulatory gene. So regulatory gene actually just controls the structural gene and is going to be there either to tell it to make proteins or not to make any proteins. Let's say, for example, here we have got two genes, A and B. So before the genes, which, so let's say the A and B will actually be proteins, okay? Will be proteins. So before we form those proteins A and B, what do we have to do? We have the regulatory DNA that is going to control the genes that will form those proteins. So the regulatory DNA that we are talking about is going to have the regulatory gene and the regulatory sequence. Okay. Regulatory gene and regulatory sequence. But here I've just written the gene. A bit in front here also going to have the regulatory sequence. Sequence, you know, is just a group of nucleotides like cytosine, guanine, just a group of nucleotides like that. So what is happening is that the regulatory sequence is just a few bases behind the many protein coding gene. So here where I've put A, you have got a protein coding gene. That's why we're calling it a structural gene. So protein coding gene. And then some few hundred bases before it, a few hundred bases before it, we have got the regulatory sequence. Okay, there. And the regulatory sequence and regulatory gene is forming up what we are calling as the regulatory DNA. Okay. So what is going to happen is that, so as we have said, the regulatory gene is just a few hundred bases behind the many protein coding gene. And the regulatory sequence is a site of attachment of RNA polymerase. Do you remember RNA polymerase? RNA polymerase is the enzyme that is going to be forming RNA from DNA. So where does RNA polymerase bind? It is binding on the regulatory sequence. Let me write it. RNA is actually specifically RNA polymerase 2 that forms the DNA. That forms the RNA. So RNA polymerase is binding on the regulatory portion or regulatory sequence. Okay. So when the RNA polymerase enzyme gets to bind there, then the information on the genes A and B, those two I've written, are now going to be converted into mRNA. In what process? That is a transcription. Okay, in transcription. The main regulatory gene, which is this one that you can see. So we have got a regulatory gene and regulatory sequence where RNA polymerase will bind. So the main regulatory gene is going to be also behind the protein coding gene. Like this structure gene is the protein coding gene, some thousand bases behind. Okay. So the genes will synthesize a protein which is known as a regulatory protein. So what do the regulatory genes do? Will synthesize regulatory protein. Even from the word, this is a regulatory protein. What is going to be now the function of this regulatory protein? Is now going to come on top here to control protein synthesis. So it's now the one which is going to be giving instructions to say, okay, you can form proteins. Actually, you are forming them at a slow pace. Can you speed up the rate at which you are forming the proteins? Or the process is too fast? Can you reduce? So that is what is going to be happening. The regulatory protein, when it is being produced, it is going to go on and control the binding of the enzyme RNA polymerase. Are you following how the steps are going? So the regulatory protein has been formed by the regulatory gene. It's now going to tell RNA polymerase to say, okay, RNA polymerase, you can now bind to the regulatory sequence. Okay. And when the RNA polymerase does bind to the regulatory sequence, RNA polymerase can either increase or decrease, okay, depending on the situation. So if it increases, it means that you need to form more mRNA, right? Because when you form mRNA, it means now you are going to form the proteins which are being encoded for by those structural genes A and B. Okay. So When we are talking about a Lac Operon, so this was just to help us understand about the Lac Operon. So what about the Lac Operon then? So the Lac Operon is a group of genes that regulate the metabolism of lactose. You know, where is lactose found? So group of genes that regulate metabolism. And you know, metabolism involves catabolism and anabolism, the making and the breaking of a compound. So the group of genes that regulate metabolism of lactose is what we call the Lac Operon. And you know lactose is a sugar that is made up of galactose and the okay, I wanted to say galactose and glucose. So let me just mention the three big ones. So the three big disaccharides, lactose, you have got sucrose, and then you have got maltose. What are they made of? So maltose is glucose plus glucose. Sucrose is glucose plus fructose. Okay. And then lactose, this one is galactose and the glucose. Now I've written GG and GG, but here there is a galactose. Maltose is two glucose molecules. Okay, so now that we know what Lac Operon is, so when we talk about one bacteria which is known as the E. coli, Escherichia coli, the E. coli bacteria is actually normally found in our bodies in the GIT, the gastrointestinal tract, the one used to call digestive system. You are going to have the E. coli, and this E. coli is normal. Normally, when you have got bacteria in your body, those bacteria are going to cause infections. They'll multiply and then they are going to start spreading and they are going to affect the normal balance of the body. But then E. coli is normally found in our body, meaning that it doesn't cause any disease. It's actually going to help us in the breaking down of some things. So glucose we know is used as an energy source. So for the same E. coli bacteria that we are talking about, glucose is going to be the energy source. So most the E. coli does not use the lactose. Okay, so we are talking about E. coli. So E. coli is a normal resident bacteria. It's found in our GIT. Okay, so normal resident bacteria in GIT or the gut tube. So this E. coli, we are saying that what does it utilize if it should work, it uses glucose as a source of energy. So how about, let's say you have drank milk which contains only your lactose. What are you going to do? Because you are not using lactose as your energy source. You are using what? Glucose. So what is going to happen is that you are going to break down lactose. So let's say for a milk meal, what is going to happen if you get a milky or milk meal? You are going to get this milk or the lactose which is found in milk. You break it down. Okay, you are going to break it down into glucose plus galactose. And what is helping in the breakdown to glucose and galactose is what? So what is helping in doing this is the Lac Operon. So that is why this guy is very important. Otherwise, if this bacteria isn't fit, is if it doesn't have any glucose, it means that it will not be able to help. Actually, these bacteria, we call them normal flora, meaning it's normally found in our body. So if it is not there, if there is any bacteria that is going to come to our body that is not part of our body, is going to cause infection. So in other words, the E. coli competes with harmful bacteria so that that bacteria doesn't get to grow in our bodies. Okay. So as we have said, when we get to eat food or milk that has got lactose, lactose is going to become essential for the energy of the bacteria in the absence of glucose. But it's not direct like that because we have to break down this lactose. So the Lac Operon has got different segments. So let me try to draw it. So as we have said, Lac Operon is a regulatory gene. It's controlling the amount of protein that we are forming. Okay. So remember when we are talking about this regulatory, the gene regulation, we have got the regulatory portion and the structural portion. Let's say the regulatory portion is this one. So this portion is going to be controlling the amount of protein that we form. We are also going to have the what? The structural portion or the structural gene, the one that is actually coding for the proteins that we want that will be able to break down our lactose. But when it comes to the Lac Operon, there are three important structural genes that you need to know. Only three. The first one is known as Lac Z. The second one is known as Lac Y. And then the third one is known as Lac A. So these are the three structural genes that you have to understand. So what is a Lac Operon? Lac Operon is a piece of bacteria DNA which is going to be made up of few genes which are very important to help in the breakdown of lactose. Okay, it is found in DNA, I mean in bacteria to be specific. In this case, we're discussing E. coli. So the Lac Operon, we can divide it into two important regions, the structural portion and the regulatory portion. The structural genes will code for enzymes which regulate protein, which will be able to regulate the control of structural genes. Have you gotten that? So the structural genes, we are going to form what? Will form enzymes. So this guy will form enzymes. But those enzymes that we form, what are their functions? So those enzymes will regulate the control of gene regulation. Form enzymes which regulate control of gene expression or which regulate gene expression. Okay, basically that. And when we are talking about the regulatory gene, the regulatory gene can either be on or off, and that's the most important part of this topic. Okay, it can be switched on and off. So you can forget many things, but don't forget this. Actually, the main instance of this class is just to understand how we can switch on and off the structural gene. If you switch it on, it means you now make your proteins. If you switch it off, it means you don't make your proteins. Okay? So as you can see, we have written three genes there. But before I talk about those genes, what is the function of the regulatory portion? The regulatory portion is actually going to be divided into Lac I, which is going to code for a protein, which is known as repressor. Okay? The operator, the promoter, and at the end of the Lac Operon, a terminator. Those are some things made up which are going to found in the regulator portion. So what does the regulator portion has? It has, let me list the genes which are present in the regulator. Number one gene in the regulator, you are going to have lac I, lac inducible. The, this one, the lac I, what does it do? It forms, it's going to code for a protein, codes for a protein known as repressor. This is a very important one. Okay, very, very important protein. So that is the function of I. What other portions do we find on the regulatory portion? We have got the operator. What other thing are we going to find? We have got the promoter. Is there anything else? At the end, we are going to have what we call the terminator. They will ask you this in the exam, which of the following make up the regulatory portion? Like I, operator, promoter, and terminator. Now, the three genes that we've mentioned on the structural gene, you need to know the order in which they are. It's like Z first, like Y second, and like A. Even if these four that I've written here, you are going to know which one is the first one. Okay, but then let's talk about the like Z, like Y, and like A. What do these form? So starting with lac Z is going to code for the enzyme beta galactosidase. Beta galactosidase. Okay, so that is the enzyme that we are coding for by lac Z. Now, what is the function of beta galactosidase in the breakdown? Remember this process we are discussing, the breakdown of lactose. So beta galactosidase is going to break down the bond between glucose and galactose, even the word galactosidase. So this one will break the bond between glucose and galactose, and so you convert lactose into its simple sugars. So in other words, this guy is the one that is getting the lactose and then converting it into glucose and galactose. Okay, so this is important, don't forget that. How about lac Y? What is its function? Now, lac Y is going to code for a protein which we call beta galactoside permease. Beta galactoside permease. Again, you need to know what is the function of the beta galactoside permease. Even from the word permease, what do you think that means? It is for permeability. So it's going to fit in the cell membrane of the bacteria and allow more lactose to be pumped into the cell. allow lactose pumping. Okay, so I want to pump lactose into the cell. Remember, you've eaten or you've drank your, let's say, your milk, right? The lactose is just in the GIT, it is in the digestive system. It has to enter our cells. So that is a function of beta galactoside permease. It will allow the lactose to enter the cells in there. That's where it is going to be broken down to glucose and galactose. How about Lac A? Lac A is going to code for the enzyme beta galactoside transacetylase. Okay, beta galactoside transacetylase. Okay, so basically these are the three enzymes that you code for. Please make sure that you know them. Now, as we have said, the regulatory portion is made up of like four parts. Okay. So what is going to be happening is that the enzyme RNA polymerase is going to attach to the promoter, which is behind the operator. Now, let's talk about the four proteins that we have talked about. I mean, the four genes that we've talked about on the regulatory portion. Like I, operator, promoter, and the terminator. How are they? So I'm going to write this again. So here we are going to have like I, and then we are going to have the promoter. Okay, and then we are going to have the operator. These are the ones I talked about, and I said that at the end, that's where we're going to have the terminator. The terminator is at the end there. And so these are the structural, these are the regulatory genes. Remember, you also have structural genes, right? The ones which are actually forming proteins. What are the regulatory genes that we mentioned? The structure genes we mentioned? We have got Lac Z, and then Lac Y, and then Lac A. What is Lac Z coding for? For the protein beta galactosidase. Lac Y, beta galactoside permease. Lac A, beta galactoside transacetylase. Okay, so remember what we said in the beginning. We said that the enzyme RNA polymerase is going to attach where? To the promoter. Okay, please take note that the promoter is behind the operator. So that is where the enzyme is binding. Okay, so here you can see this guy. This is RNA polymerase. What is the function of RNA polymerase? It is to make RNA from DNA. But then, what is going to control the function of RNA polymerase is the operator. Okay. So, what is the function of the operator? It is a switch. So if the switch is turned on, it means that RNA polymerase will now go forward. You know, if RNA polymerase goes forward, it means that it's now going to cause the Lac Z to produce beta galactosidase, like Y to produce beta galactoside permease, and like A to produce beta galactoside transacetylase. But if the operator is off, it means that RNA polymerase cannot move forward. That is very important. Okay, so in the RNA polymerase, when the operator is off, RNA polymerase cannot move forward, and so it cannot make the enzymes that we have talked about. But then what is going to control the function of the operator? It doesn't control itself for it to be on or to be off. So like remember the we said that I means inducible. Where can I even write it? Lac inducible. It means that you can tell it to be on or off. That's the meaning of the word inducible. You can induce it, you can tell it, okay, you can now be on or be off. So lac is going to do something. What lac does is that it's going to produce a protein. I mentioned that protein. What's the name of the protein? Repressor. Okay, so this one is going to produce or to synthesize repressor. And what is going to be the function of the repressor? Repressor is going to come and hold the operator. So it is going to produce, what is it producing? This guy is known as the repressor. Now look at this, look at what is going to happen. So when lac produces repressor and the repressor is held on the operator, that is going to mean you are in the off state. Okay, so what is the off state? Off is when repressor is bound to the operator. And then off or on, to switch on the operator, the repressor is out, is not bound. But what is going to cause the repressor not to be bound to the operator? It is the presence of lactose. That is one important thing. Okay, the presence of lactose. Okay, again, repressor 90% of the times you are going to find it is bound to the operator. In other words, the repressor most of the times has switched off the operator. But then there are two conditions. When you have got lactose, okay, and you don't have glucose, take note of the two conditions: presence of lactose and the absence of glucose. Because if you have got lactose but you also have glucose, this gene is still going to be off because the glucose is going to be used by the bacteria, not until you finished all the glucose and then you have got lactose. That's when you can now switch on. Okay, so how do you switch on the operator? What is going to happen is that lactose is going to come. Let's say this is lactose. Here is our lactose. When this lactose goes and binds here, it means that is going to make the bond with the operator to be weak. So what is going to happen is that those, can I call them legs or something? Okay, so the hands which are holding are going to be off. So you see the molecule is going to be something like this when lactose is here. So the presence of lactose on the repressor is what makes it to get out of the operator. So this is lactose, and this is the repressor. So when that happens, so when the operator is free, you can see now the operator is free. When the operator is free, what is that going to communicate? It's now going to mean, okay, RNA polymerase, move forward now. And so RNA polymerase is going to move forward, and then it's going to make our proteins that we talked about, the beta galactosidase, beta galactosidepamase, beta galactoside transacetylase. The bacteria can only produce these three enzymes when you have got lactose and you don't have any glucose. Now, sometimes actually behind the promoter, you have got a small protein which is important. So behind the promoter, just here, you have got a protein which is known as the CAT protein. What does CAT mean? Catabolite activator protein, CAP. So you have got CAP. What is CAP? Let me write it here. Catabolite. No, catabolite meaning it's involved in breaking down. Catabolite activator protein. Okay. So yeah, we have got the cap behind. What is going to happen is that this cap helps RNA polymerase to hold to the promoter so much more. Actually, I've placed it at a wrong position. Okay, okay, this is fine. It is behind the promoter. So what is the function? Help the promote help RNA polymerase hold to what? To the promoter. That is the function. Okay, more effectively. And so if it holds more effectively to the, what would be the importance of RNA polymerase holding more effectively to the promoter? It's going to increase the rate at which the enzymes will be able to transcribe the DNA. Okay. So the genes of Lac Operon are ultimately going to produce enzymes which are going to be used in the digestion of lactose. Now, cap protein is not always active. Remember, if it's active, it's going to make your lactose, your RNA polymerase to be held to the promoter, but it's not always active. That's how it gets its name as catabolite activator protein, because it's only activated by catabolism. If there is need to break down lactose, that's when it's going to be activated. Otherwise, if there is no need to break down the lactose, there is no need to be activated. So, but how does it do that? So how it does it is that if glucose and galactose are both present, first the bacteria is going to use up all the glucose inside the cell. But when the glucose gets low, it's going to be now a signal to the cell and the cap protein to be activated. So what will activate the cap protein is there is low glucose. And the activation of the cap protein is going to be via cyclic adenosine monophosphate, CAMP. So CAMP is what will lead to the activation of cap. Okay, so that is what it does. So whenever glucose concentration in the cell gets to drop, what will happen is that cyclic adenosine monophosphate is going to rise. Why does the cyclic adenosine monophosphate rise? That is because when glucose has dropped in the cell, hormones are going to be released. Okay, like the hormone glucagon is released, and that is going to go via, is going to produce CAMP. That CAMP is going to do what? Is going to cause the cap protein to be activated, and when the cap protein is activated, is going to bind to the cap site. So I've got a cap protein and the cap site. So the cap protein is going to be produced, and then it binds to the cap site, where, which is this site here. And when it does that, it's going to help RNA polymerase to increase the rate of transcription. So this, my friends, is what we call the regulation of gene expression. So there is also something that is known as the end product inhibition. So let's say, for example, we have got A causes the production of B, B causes the production of C. So end product inhibition means that when you have formed the C will now go to the first thing, which was A, and then it deactivate it. When it deactivates, then it means that you can't make B again, and you will not make C. So that's the end product inhibition. So let's do some of these questions, and then we are done. The term gene expression is the production of a protein from information on DNA. Is that true? I can put a dot there, but we don't produce proteins from information on DNA. The information actually comes from what? From RNA. But the instruction of first come from DNA. B, production of gametes from the male and the female. That's not true. Fertilization of the female gamete. That's not true. Conversion of an enzyme from its inactive form into its active form. No. Removal of introns from pre-mRNA. No. So the best one we can actually pick is this same one. Okay, production of proteins from information on DNA. So remember, it involves a lot of steps. Choose the item which is not part of the Lac Operon. Repressor, okay, regulator, inducer, promoter. You know the promoter is part of the Lac Operon, right? What else is part of the Lac Operon? So you have got the Lac I, the promoter, the operator, you have got the terminator, and then those structural genes. Actually, let me even show you this diagram now. So you can see we have got the cap protein here, you have got Lac I, and then you have got the promoter Lac P, you have got the operator Lac O. These are the regulatory genes. How many are they? They are like 4. The cap, okay, the regulator, the promoter, the operator. And then you have got the Lac Z producing beta galactosidase, Lac Y producing lactose permease, Lac A producing beta galactoside transacetylase. Here they've put thiogalactoside transacetylase, right? So that is it. You can see regulator, promoter, operator. So the regulator is actually also part, even the repressor is part of the Lac Operon. Remember, the repressor is produced by Lac A, right? But we don't have the inducer, do we? Okay, so that is the answer that we are going to choose. But when you look at the repressor necessarily is not part of the Lac Operon, but is produced by Lac I. So here actually the best answer would be 5. So repressor is produced by what? Lac I. It's not like it's part of the Lac Operon. Expression of gene in a cell depends on the type of cell and its function. This is true. Remember, we said that depending on the cell and also its function, you can decide to form genes or not. Its biochemical condition, yes. So if conditions are right, you can now get to form. It's a cellular signal, yes. So if you tell the cell to say, okay, you need now to make genes, you do it. Presence of appropriate enzyme in the cell, Yes, actually enzymes are going to be there to activate the genes. So all of the above are correct. In a prokaryotic cell, they precedes the dash structure operon. In a prokaryotic cell, the dash precedes the dash structure operon gene. So I don't know what the question was trying to say. In prokaryotic cells, they precedes the dash structure operon gene. Okay. Lactose molecule, repressor, promoter, operator. Anyway, you can just pick 3 and 4. I don't know whatsoever they are saying here. Unless if there is the dash that they didn't mention. Okay, but you know that the what comes about to precede the operator and the promoter are going to come in front of the structural gene. Okay, remember the structural gene is in front, and then you have got a promoter and the operator behind the structural gene, isn't it? So that is what we just mentioned. Which of the following is true of the Lac Operon in E. coli? The operon is only switched on in the absence of lactose. Is it absence? No. The Lac Operon is polycitronic mRNA. So the meaning of polycitronic, if we can remember from what we did discuss on transcription, what does polycitronic mean? Okay, so when you're talking about, there are some genes that can form more than one end product, like they can code for more than one thing. That is the polycyclonic. And then you have got those that can only form one or code for only one thing. That those will be the monocyclonic. Okay, so basically that. Just a moment. All right. Yes, so as I was saying, so eukaryotic transcripts, if it's in a eukaryotic cell, tend transcripts tend to be monocyclonic, meaning that they have one gene per transcript. So one gene is going to tell you one transcript or one thing that you are going to form. Okay, that's for eukaryotic transcripts. And then for prokaryotics, these ones will be coupled. They are going to be like polycyclonic. Okay, let's look at the other options first. Beta galactosidase is only produced in large quantities when the Lac repressor is bound to the operator. The repressor shouldn't be bound. Operon enzyme transcribe the structure genes when the Lac repressor is bound to it? No, not when it's bound. And then 6 is only saying this one was actually supposed to be continuation, is bound to the operator site. It's not supposed to be bound. So the answer is 2. Okay. So the Lac Operon is polycyclonic in all prokaryotic cells, meaning that more than one gene, right, having to code for a transcript. Actually, a gene for coding for more than one transcript. The role of the inducer that regulate a repressible operon is to the role of an inducer that regulate a repressible operon is to bind to the promoter region and decrease the affinity of RNA polymerase, bind to the promoter region and block the attachment of RNA polymerase, increase the production of inactive repressor protein, bind to the repressor protein and activate it, bind to the repressor protein and inactivate it. So the role of an inducer, okay, that regulate a repressible operon. So an inducer, I've not really mentioned about this one. Okay, so let's talk just a bit about it. So inducer in Lac Operon, what is it going to do? Okay, so the inducer in this case, what is going to happen is that it's going to bind to the repressor. When an inducer binds to a repressor, it's going to cause it to change, okay, and to be released from the operator region. And an inducer that we can give an example is lactose itself. Lactose, when it binds to repressor, the repressor is going to be on now. The repressor is going to move out of the operator. So that's the function of the inducer. Okay. So it will be able to bind it not to the promoter, not to the promoter, increase production of inactive repressor protein, no, bind to the repressor and activate it. No, is to bind to the repressor and inactivate it, because when the repressor is out of when the repressor is out of the operator, it is off. Okay, it has been inactivated. The way it is bound to the operator, that's its active form. In the function of the Lac operon in E. coli, the Lac genes are transcribed only in the absence of glucose, because glucose is an inducer, a regulator, not needed by E. coli, needed in the breakdown of lactose, preferred energy source. Yeah, glucose is a preferred energy source. So if it is there, you will not be able to break lactose. An operon is a or an RNA sequence made up of codons that work together, group of proteins that work together. It's not a group of proteins, group of amino acids, not amino acids. Double-stranded DNA sequence of nucleotide, really not. Single-stranded DNA. So it's RNA sequence made up of codons that work together. Okay, they are working together so that you can form something at the end. Choose the item which is not part of the operon. We've already, haven't we answered this one? Repressor is part, operator, promoter, and structure genes. So it is a regulator. It's the same thing as we did here, isn't it? Okay, which one was that? Okay, choose the item which is not part. Okay, so for this one, regulator is going to be part, so it means it's only the inducer, which is not part of the Lac operon. So forget about this one. It's going to be the inducer. This is how you can even manage answering some questions, because here there's no option that is picking to, isn't, you can see. So the regulator is not part. That is why for also this one, the regulator is okay. So for this one, even the regulator is going to be part of the answers. No, the other one is saying not part. This one is saying not part. That one is, so they are a bit different questions. So these, the regulator and the inducer. The inducer, we said, is not part of the Lac operon, right? But think of the, let me just make this question a bit clear. Inducer will include lactose or anything that will come and bind to the repressor. We want what is not part. Repressor is part. Promoter is part. So you've seen this option 5, you're saying 1 and 3. 1 is already part of the answers, so it can't be, right? So the answer here is regulator. The inducer is part. The inducer includes lactose, which is going to come and bind to the repressor. So it's part of the Lac operon. So unless the regulator, okay, will not include it. Okay. In an operon, the promoter site codes for the repressor protein. Is that the function of the operator? No. Is where the repressor binds? Yes. Is where RNA polymerase binds? No, it binds to the what? Promoter. Codes for the regulator, so we've already seen the answer. Identify a protein that is produced by regulatory gene. Beta galactosidase? Yes. Inducer? No. Transacetylase? Not just transacetylase. Beta galactoside transacetylase. And then repressor? No. Permease? No, it's beta galactoside permease. The Lac Operon is a group of genes involved in the metabolism of what? It's lactose, isn't it? The Lac Operon is switched on when replacer molecule binds to the operator. No. Glucose is present. No. Lactose is absent. No. RNA polymerase binds to the operator. No. Glucose is absent. Yes. What is the function of the permease enzyme in the Lac Operon? Permease. Hydrolysis of lactose? No. Attachment of lactose to the Lac Operon? No. Breakdown of lactose? No. Entry of lactose into the cell? Yes. Lac Operon disassociates from the operator site when it binds to. Lac Operon disassociates from the operator site when it binds to lactose, operator, repressor, glucose. Okay, this is not Lac Operon. I've been reading it wrongly. Lac repressor. When does it disassociate? It's when it binds the lactose. In an operon, the function of the promoter is to code for the repressor. No. Bind the repressor. Yes. It doesn't code for regulatory genes, it doesn't bind RNA polymerase. This is for the promoter. Okay. So it's bind to the repressor. Choose the item which is not part of the Lac Operon. Inducer, we've said it's part. Operator is part. Promoter, structure genes. So it's the regulator, right? But then I'm supposed to visit this question again. Which one do you think is not part of the Lac Operon? Is it the inducer or the regulator? Actually, the regulator is part of the Lac Operon if we just consider it well. What is not part is going to be the inducer. I'll explain this just in a moment. Let's just answer these others. What is the function of beta galactosidase? Entry of lactose? No. Synthesis of lactose? No. Attachment? No. Breakdown of lactose? Yes. Repressor becomes inactive when it binds to? Yes, when it binds to lactose. Remember, when it binds to lactose and then it's released from the operator. What is an operon? Part of a ribosome? No. Part of a RNA? No. A unit of made up of linked genes that code for proteins needed for specific tasks. Very good definition. That's it. A special gene used in regulation of gene expression. A set of unrelated genes located close by a chromosome. No. So it's a unit of linked. They are linked genes. They are close. Okay, so that is it. Now let's come back to this question. We answer it. So between inducer and regulator, which is not part of the Lac Operon, it's actually supposed to be the inducer. Okay, the regulator is part of the Lac Operon. So that is going to be the same with which one? This or not this one? Even this one. Choose a statement which is not part of the Lac Operon. So we're talking about inducer and regulator. So here we have got repressor. The regulator is supposed to be part of the Lac Operon. But how about the repressor? Again, so if on the other question we chose the regulator, it means that even he is supposed to be part of the answers. Okay. And then the other first question that we did answer. Yeah, this one. Or not this one. This one. Choose a statement which is not part of the Lac Operon. Repressor, regulator. But here now you have got both regulator and inducer. Which one would you pick? So inducer is not going to be part of the operon. Okay. So let's make sense of it using this diagram. Can you see that we have got a regulator here, which is the Lac I? It's Lac I which is producing the repressor. So in that case, I would say the repressor is really not part of the Lac Operon. But almost all the things which were mentioned were part. Lac Operon has got structural genes. It has got the operator, the promoter, and the regulatory gene, the terminator region. Even a repressor protein is part, even an inducer. But then inducer is not going to be really part of here, but it's going to come from the outside environment to come and bind to the repressor. The repressor itself also is not direct from here, it's actually just produced from Lac I. So best if you have got those options, make sure that regulatory you always choose it, the promoter always choose it, and the operator always choose it. The others that you can have, you can decide not to. All right. So basically that is it.